have high levels of enzymes involved in fixing nitrogen. That's their primary function. That other cell is an akinete. Now, akinete, remember I said A or N in front of a word means without? And what's kinesis? Movement. So akinete refers to something that is not moving, but it applies more to not the cell moving through the environment, but things not moving within the cell, because an akinete is a dormant structure. They're not as resistant to environmental factors as endospores are, or exospores, but they are nevertheless resistant. So they can resist extreme low temperatures of winter or the hot, dry temperatures associated with summer. So these are resistant cells, again produced by cyanobacteria. Akinetes are larger than heterocysts. They tend to be somewhat long. So here you see an akinete, much larger than the vegetative cells. These are vegetative cells. This is a heterocyst. Sometimes akinetes and heterocysts are adjacent to one another, but not always. And that's another thing that we can look for when we look at the cyanobacteria prepared slides. So akinetes basically allow cyanobacteria to survive periods of unfavorable conditions. Cyanobacteria are often found living in water. If they're living in water and the water freezes, that's not a nice condition for cells to survive in. Their metabolism is basically going to stop. So akinetes can survive that extremely harsh cold of winter. During the summertime, think about what happens to a lot of water habitats, especially here in California. Things dry up and they become exceedingly desiccated, and they're exposed to ultraviolet light and they're hot. And so akinetes allow cyanobacteria to survive that way. So please don't talk during the lecture. It's very disturbing. Now, conidia. This is the spore type that is a reproductive structure. These things are called conidiospores or conidia. The root word is conus, which means dust. These are tiny little structures. They're reproductive structures, and they're only produced by a certain group of bacteria that have a filamentous morphology. So these bacteria form not cocci or bacilli. Their cells are long strings. They look more like spaghetti than anything else. And at the ends of these long strands of spaghetti-like cells, there will be a series of small units. They can be cocci in shape or they can be bacilli in shape, and these are conidia. So here we see the diagrammatic representation. The filamentous bacteria, again, long strings and often branching. Here are the conidia at the end. This is an actual photomicrograph showing conidia. These are not quite so cocci shaped. They're more like bacilli. These you can't see the filament they're attached to, but that's how those things form. Bacteria that form conidia, if you're looking at the colonies growing on an agar plate, the surface of the colony will look like powdered sugar. It looks distinctly powdery, and you touch it with a loop and it comes off as powder because those are the conidia. So these are reproductive structures. Each one of those little balls can form a new cell. The only bad part about that, as I said, sometimes people will talk about bacteria producing spores, and you need to know what kind of bacteria are the people dealing with, because if they're dealing with Streptomyces, and they say the Streptomyces is producing spores, they're talking conidiospores, not endospores. The last specialized structures are spiroplasts and protoplasts. These are just cell types that are formed if for some reason bacteria lose their peptidoglycan. So as I said, peptidoglycan is a normal, rigid material. Is everybody here? We're missing a person. I'm going to unlock the door. Otherwise, he can. Not even the anatomy students making noise, it's the anatomy instructors. Okay, anyway, spiroplasts and protoplasts, they form when the bacteria lose their peptidoglycan. The difference between them has to do with what kind of cell was there initially. If the cell is a spiroplast, it initially was gram-negative. And a spiroplast, when it loses its peptidoglycan, it still has the outer membrane of the cell wall. Remember, gram-negative cells have a cell membrane, and then a thin layer of peptidoglycan, and then an outer membrane. You take away the peptidoglycan, you still have that outer membrane. A protoplast is made from a gram-positive cell, and it has no wall at all. These things are particularly sensitive to changes in osmotic pressure. They don't have a wall to protect them, so if you put them in a hypertonic environment, they will tend to blow up. And that is the end of our prokaryotic cell presentation. That brings us to the topic that we're supposed to be covering according to the schedule. So we're talking about taxonomy. Taxonomy and classification. If you look up the word taxonomy, it has different meanings depending on what discipline you're talking about. So taxonomy, when it applies to the biological sciences, is a little bit different than if you're talking about English, for example. For us, taxonomy is the science or study of the classification of organisms. Typically, because we're biological science and biology, bio means life, we think of categorizing or classifying living things, but taxonomy can also apply to non-living forms, things that used to exist. So even though it says living organisms, fossil forms also have been categorized using taxonomy. Basically, what this involves is dividing organisms into groups, categories if you will, but dividing them into these groups require that there be specific criteria that are considered. What are we basing our divisions on? We're going to categorize things. What are we going to use to categorize, to put things in categories? And taxonomists determine that. They try to look at organisms and they try to put them in categories showing natural relationships between organisms. But as you're going to see tonight, this business has changed quite a bit, and part of the change has to do with what we know about organisms. What have we learned about organisms over time? So we try to decide what are the criteria for putting things into each group, and the criteria keep changing. This is a section of biology that causes people a lot of trouble. For some reason, a lot of students don't like taxonomy. And yet, if you think about it, people categorize things all the time. We're always putting things into categories. I think it has to do with the way our brains work. We try to categorize. I can give you some examples. Let's say we were talking about modes of transportation. You can relate to the fact that there are multiple different modes of transportation, and we can kind of throw these up there and they bring to mind certain things. You can travel by automobile, plane, boat, train, spaceship, well not yet. In the automobile category, we have subcategories: sports car, SUV, compact car, luxury car. Under compact, we could subcategorize again, foreign versus domestic. And then under foreign car, we could say, well, we can categorize those by make, a Toyota, a Honda, etc., or we could categorize based on color, red, blue, green, what have you. There are different ways to categorize, but we do it all the time. My guess is if you went to your house, if you went into your kitchen, for example, it would be pretty uncommon for people to live in a kitchen where they go in there and just randomly throw stuff wherever. Instead, things tend to be categorized. Over here we have a drawer that has silverware in it. Up here, this section of the cupboard has this kind of plate, etc., etc. We categorize. It's something we do. So, taxonomy, as it applies to biology, again, is involving this categorizing things, putting things into categories, and determining the criteria that we're going to use to put them into these categories. This is influenced by past experience. It's influenced by how much we know, what kinds of things we know, what kinds of equipment we have, all those kinds of things. Some criteria that have been used for categorizing living organisms include, one, considering function. For a long time, protozoa, which are the animal-like single-cell organisms, they were categorized based on how they move. So it was a function. If they moved by cilia, they were ciliates. If they moved by flagella, they were flagellates. If they moved using pseudopodia, they were sarcodina. And if they couldn't move at all, they were sporozoea. So there were these categories based on how they function, how could they move around. Another way to categorize is to consider the structure. What's it made of? Does it have glass in its cell wall? Does it have calcium carbonate in its cell wall? So in order to categorize based on structure, oftentimes you have to do a little bit of investigation. Like we talked about peptidoglycan being the wall material of all bacteria. You can get more detail. What kinds of amino acids are in there? You're looking at cell membranes, what kinds of fatty acids are in there? So you can really get into the molecular structure of organisms. Another way to look at it is just, what does the thing look like externally, morphology? This is useful for plant taxonomy. Anybody who's studied plant taxonomy, you know that there's a lot of dissection of flowers, and you count flower parts, how many petals, how many sepals, how many stamens, etc., etc. So it's based on what the thing looks like. Well, these are all very useful until you get to organisms that are single-celled. And it's like, ugh. Well, mostly that's what we deal with, bacteria. So there are categories, or there are criteria for establishing categories. Again, they're supposed to show natural relationships between organisms. So if we put organisms in the same category, they should be related to one another on some basis. So the criteria have to deal with that. What is the basis we're going to use? The other thing about taxonomy is taxonomists try to put organisms into categories that show phylogeny. This is really tricky. Phylogeny is evolutionary history. What kind of organism gave rise to another kind of organism? This has changed quite a lot over time, again, because we have different ways to investigate organisms. So phylogeny is evolutionary history. And as I said, the criteria have changed. Taxonomy itself has changed extensively. So we're just going to kind of look at the basics here. There have been some very important contributions. For one thing, if you're going to divide organisms into categories, you have to have some way to identify each organism. So the identification of each organism currently involves what we call binomial nomenclature. Binomial nomenclature means that each organism has a two-part technical name. Binomial means two parts, and nomenclature just means you ought to have a name. So a two-part technical name. This system was developed by Carolus Linnaeus. As soon as I put that up there, and then you'll notice he was a Swedish naturalist. Carolus Linnaeus is a Latinized form of that person's name. So that was not his original name, but his name is Latinized in the manuscript. He published a manuscript in 1735, and in this manuscript, he gave specific descriptions of a whole series of different types of organisms. And then off in the margin, he would have a two-word short version of this description. So for example, a robin might be described as a thrush-like bird. It has a red-colored breast and a gray back, and it has such and such. It's got a little bit of an eye ring, a white eye ring around it. It's got a black head. So there would be this long description of it, but then there would be a two-word summary. The two-word summary in this case is Turdus migratorius. Turdus means it's a thrush, and migratorius means it migrates. So this name tells you something about the organism. This was a thrush-like bird. It is migratory. That two-part name, then other biologists picked this up, and they liked the idea of having a simplistic two-part technical name. And so it became established as this is how we're going to name things. So in binomial nomenclature, this two-part name, the first part is the genus name. Genus is a taxonomic rank or category. The genus name is always capitalized, and it's followed by a specific epithet, which basically means species name. The specific epithet is not capitalized, so it's all lowercase. So the genus name should be capitalized, the specific epithet is not. Both of these are Latinized. Sometimes they have Latin roots, sometimes they have Greek roots. Sometimes terms are based on an individual that may have made a discovery. You've undoubtedly heard of Escherichia coli, E. coli, what people call it, but E stands for Escherichia as opposed to electronic. It's not like email. Anyway, Escherichia is named after Dr. Escher, and I believe he was German. So it's not Latin or Greek root, and you'll see this quite a bit. Anyway, these terms are Latinized, and you might think, well, was that because Linnaeus was an elitist? No, it was because Latin was the language that was used in educational institutions at the time that he was putting together this manuscript. That was the language that was used. It was handy because this is not a language that's spoken, except in the Catholic Church and in science. So it hasn't changed. It doesn't keep changing like other languages do. By convention, the genus is capitalized, the specific epithet is all lowercase, and both are underlined. So when you get your quizzes back this evening, you're going to see that if you put Streptococcus pneumoniae as the etiological agents, the most common etiological agents for pneumonia, if you didn't underline it, I did. That's how it is supposed to be. And I think I told you guys that. Actually, on the first quiz, I wrote it on there. Please underline technical names. It's convention. That's how it's done. If you are writing it by hand, you underline it. If it is in print, it's italicized. That has changed somewhat over time, and it had to do with typesetting, and then it had to do with computer fonts. Now it's easy. Just italicize it. If it's a technical name, put it in italics. Now, as I said, these binomials are short versions of descriptions, but they still tell you something about an organism type. For example, if you have spherical cells in grape-like clusters, the term for that is Staphylococcus. Coccus is the shape, sphere. Staphylo means grape-like cluster. So Staphylococcus is a genus name. It is not the genus name that goes with pneumoniae. Streptococcus and Staphylococcus, two very different genera. The specific epithet, aureus, means golden colored. They are gold colored depending on what medium you put them on. This one appears to be on blood agar, so the gold is probably carotenoid pigments the cells are producing themselves, but not all Staphylococcus aureus produce gold-colored colonies. You may have noticed on our mixed A plates, ours are white. Nevertheless, Staphylococcus aureus is a binomial name. It's a two-part technical name for a particular organism type. Now, what's really important about this, as I said, Linnaeus developed this early on, and he was putting together a manuscript and just kind of put these little short versions out there. But now, the technical names allow for communication throughout the world, and it doesn't matter what language somebody speaks. If they turn in a publication, the technical name should be the same. A manuscript can be written in German. The technical names are going to be the same. They're in Latin. You publish in English, the technical names are still in Latin. You publish in Russian, Chinese, Italian, it doesn't matter. The technical names are Latin, and they stay the same. So this is important because it allows for communication between scientists throughout the world. So please always write technical names correctly. Genus name capitalized, specific epithet not, both of them underlined. If you write them by hand, italicized if they're in print. At some point, I'll start taking off points when you don't. So do it right. So Staphylococcus, genus, capitalized. Aureus, specific epithet, lowercase. Both italicized or underline if you write them by hand. Okay. The genus and the species are two taxonomic ranks. So they are two categories. When we started categorizing things, or if we look back and said, well, about the time that Linnaeus was putting together his manuscript, there were two broad categories of organisms, and then within those smaller categories, this has changed a little bit also, but a group of organisms that occupy a specific rank is called a taxonomic rank or a taxon, and these ranks, these categories are nested. So if you have one big category, let's say your big category is a bedroom. Within that bedroom, there are smaller categories. Over here, there's a dresser, and over here, there's another dresser. Within those dresser categories are drawers. And this one has 6 drawers, and this one has 6 drawers. Categories, right? The categories are nested. Each specific category is a taxonomic group or a taxon. Plural is taxa. And these are the taxonomic ranks. These have been around in biology textbooks for a very long time. The ranks are kingdom, phylum, class, order, family, genus, species. There are some mnemonic devices to help you remember them. Kings played chess on fine grained sand. Kenny Porter couldn't open Frank's gas station. Kids playing chase on freeways goes flat. Whatever works. A lot of the mnemonic devices are somewhat evil in the biological sciences, especially in anatomy. You have to memorize all those nerves, those cranial nerves. Anyway, yeah. So notice kingdom, if it's plural, you just put an S on it. Phylum, if it's plural, it's phyla. Class, put an S on it if it's plural. Order, put an S on it. Family, spelling changes, but most people are familiar with changing a Y to an IES. Genus is singular, genera is plural. So you don't talk about a genera. Genera is plural, like bacteria, plural. And species is spelled the same, whether it's plural or singular, it's species. Unless you want to call it a specific epithet. Most people don't. So kingdom, phylum, class, order, family, genus, species. From most broad category, at the top, the bedroom, down to the smallest category, some box in a dress drawer somewhere. So, at the time Linnaeus published his work, back in the 1700s, there were only two kingdoms, and the kingdoms were Plantae and Animalia. If they weren't Latinized, what are those kingdoms? Plants and animals. For us, plants, that's one of those kingdoms we rarely look at. We tend to ignore the plant kingdom. We do look at the animal kingdom somewhat, but it turns out not everything fits nicely into two kingdoms. It seemed like they did in 1735, but not so much. Once microscopes were developed and people could see these microscopic forms, things got a little more complicated. You're looking at these single cells and you're going, are those plant-type cells or are they animal-type cells? For the longest time, biologists said, if it's green, plant-like, we'll call it a plant. If it swims on its own, moves, animal type, we'll call it an animal. And so the algae were plant-like, the protozoa were animal-like, and they were incorporated into this two-kingdom system. However, even as early as 1866, there was an investigator who proposed that there should be a third kingdom. Ernst Haeckel, who was a German naturalist. He was looking at microscopic things and he said, no, really, there should be a separate kingdom for those things that don't obviously appear as plants versus animals. He suggested there should be a third kingdom called Protista. Well, it didn't take, because well into the 1960s, most biology textbooks had only two kingdoms. In fact, there's probably biology textbooks still that have only two kingdoms, Plantae and Animalia. Then there was a microbiologist by the name of Roger Stanier. He and his associates were using electron microscopes. And once they started looking at cells with electron microscopes, they noticed something very interesting. Some cells had nuclei and some cells didn't. Electron microscopes allow you to get a really fine view of the internal structures of cells, and they said, wait a minute. We can't just have two kingdoms, Plantae and Animalia. That is not going to work because we've got a bunch of cells over here that have nuclei and another bunch of cells that don't. So there should definitely be more than two kingdoms. Other people kind of noticed this as well. So in 1969, an American taxonomist, if you will, or plant ecologist, proposed that there be a five-kingdom system. This was Robert H. Whitaker, or oftentimes you'll see his name just R.H. Whitaker. He suggested there be five kingdoms. Keep the Plantae, keep the Animalia, add Protista for single-celled organisms, add fungi, because really fungi are not plants. They just don't work as plants. And then those ones that don't have nuclei, put them in a separate kingdom. They're going to be the kingdom Monera, that's what he called it. So the five kingdoms, again, Plantae, Animalia, fungi, Protista, and Monera. Monera was everything prokaryotic. Well, that sounded like a good thing for a while. And then in 1978 or thereabouts, an individual by the name of Carl Woese started looking at ribosomal RNA nucleotide sequences. Not sure what prompted this, but nucleotide sequencing had become more commonplace, and people started noticing, wow, if you use nucleotide sequences and consider nucleotide sequences, you're going to find out that there are some startling differences between cells that have nothing to do with, are they plantae, animalia, or even do they have nuclei or not nuclei. What Carl Woese found out using ribosomal RNA sequences, all eukaryotic cells are very closely related. It doesn't matter if they're plants, animals, fungi, protozoa, algae, they're all very closely related. Meanwhile, in the prokaryotic category, the ones that don't have nuclei, there were two major groups that were not related to one another at all. So that's a little startling. We tend to think of ourselves as being unique. You're more closely related to a blade of grass or the grass plant that produced that blade of grass than the two prokaryotic categories are related to one another. This was really disturbing for some biologists. Anyway, Woese proposed that there be a new taxonomic rank, and that taxonomic rank is called the domain. The domain went on top. Again, it's based on ribosomal RNA nucleotide sequences. Why ribosomal RNA? Because ribosomes are absolutely essential for living organisms. Ribosomes are the site of what? Protein synthesis. Without protein, cells don't function, because enzymes are proteins. And for that matter, RNA, ribozymes have to be, or sometimes involved in function too. So ribosomal RNA was recognized as being a very specific feature to consider. So he was looking at 16S ribosomal RNA in prokaryotic organisms, 18S ribosomal RNA in eukaryotic organisms, and found out again, all the eukaryotic organisms very, very closely related, but the two prokaryotic categories not. So there are three domains, and the three domains are archaea, bacteria, and everything eukaryotic. There's some real interesting features of this whole scheme of things, because eukaryotic organisms would not exist if it weren't for the other two categories. Eukaryotic organisms were kind of put together by a committee, and the committee was prokaryotic. And we know this for a lot of reasons, not least of all the genetics involved in these two. But also the bioenergetics, which is energy transfer mechanisms. We're going to get into that when we get to metabolism. So it's rather interesting. Anyway, these are the three domains, and again, domain is up on top, so now we've got domain, then kingdom, phylum, class, order. So if you wanted to use the mnemonic device, kids playing chase on freeways goes flat. I don't know who, it'd be darn kids or whatever. The D's gotta be fun. Those darn kids playing chase on the freeways goes flat. This is not universally accepted. Lots of biology textbooks don't talk about domains. But if you are in microbiology, we have to talk about domains because it's very, very important. The archaea, the bacteria, are not in the same category. They're very different. So there's our taxonomic ranks again: domain, phylum, class, order, family, genus, species. Now here, it would have seemed that the species or specific epithet is the most specific designation of the most specific category. Turns out that's not true either, because within species, well, if you think about dogs, for example, dogs are in the genus Canis, and the species is Domesticus. But there's Chihuahuas, and there's Saint Bernards, and there's Pekingese and Afghans, and all kinds of different types of dogs. Those are called breeds, and they're different. When you're dealing with bacteria, you can have Staphylococcus aureus, and within that category, you can have what are called strains, and the strains are very different. So you'll hear about strains of organisms, or you may hear about serological types. That's a typing based on serologies, which we'll get to in a minute. So you can have subspecies, you can have strains, you can have serological types or serotypes. Species is not necessarily the most specific category. Now, in my taxonomic scheme, where I had plantae, animalia, fungi, and then prokaryotic organisms are now divided into two domains, archaea and bacteria, a couple of things happen when the kingdom Monera is split into two separate domains. It disappears. And as far as I know, it hasn't been replaced. So there is no kingdom for prokaryotic organisms anymore, because they're two separate domains, and the taxonomy just goes domain, phylum, class, order, family. At some point, they may put some kingdoms in there, but what's not in this taxonomic scheme at all, when you think about microbes, as I introduced them the first day of class? No, the algae and protozoa are in there, so now we've got them in Protista.
Non-cellular?
The non-cellular, right, the viruses. So the viruses, the viroids, the prions, where do they fit? If you think about it, again, because we go along with the cell theory, and the cell theory says the cell is the basic unit of structure and function of all living cells. All organisms are made of cells and reproduce. Cells give rise to new cells. By that definition, living organisms are only the cellular things. What's missing is everything non-cellular. So the viruses, the viroids, prions, they still get categorized, and the criteria for categorizing viruses has changed quite a bit, because initially they were categorized based on what they infected. If you were a botanist, you studied viruses that infected plants. If you were a zoologist, you studied viruses that infected animals. If you were a bacteriologist, you studied viruses that infected bacteria. Eventually, the virologists said, hey, wait, we want to have our own set of criteria for categorizing. And so they have made their own up. We'll talk about that when we get to viruses. So viruses, viroids, prions, those guys, the non-cellular entities, they don't fit into that taxonomic scheme of things. Now, we're going to look at some criteria used for classification. There's a tremendous amount of terminology associated with this, so studying for this, again, use that study guide, pay attention to key terms. Last time in lab, we talked about morphology. Actually, time before last, because we were talking about colony morphology, and then we were talking about cell morphology. But morphology refers to external features of either colonies or cells. So morphology, again, is very useful for categorizing plants. Not too terribly useful for categorizing bacteria, because what I told you in lecture, bacteria, actually no, it was in lab, bacteria come in three basic shapes: cocci, bacilli, and spirilla. There are some bacteria that are filamentous, and I didn't mention those, and there are some odd star-shaped ones and whatnot. But if you have millions of different types of organisms and you've only got three categories of shapes, there's going to be a lot of overlap. Even when you pull in staining, you can do some things with morphology that way, and you pull in colony morphology, you're going to see a lot of overlap. You look around the room, you can say, okay, we've got colonies. A lot of the colonies that are shown here are red or orange or pink. A lot of them are smooth, shiny, circular. A lot of them are low convex. There's just a tremendous overlap when you're dealing with morphology. Again, millions of different types of bacteria, they just don't look that different. So what else can we do? This is just showing you cells, colonies. Another criteria that is used extensively when you're categorizing plants, and to some extent animals, is how they reproduce, their mode of reproduction. And as I said, when you're studying plants, if you do a plant taxonomy class, you spend a lot of time looking at plant reproductive structures, flower parts. Animals, if you study zoology, typically you spend a lot of time looking at how they reproduce, their reproductive structures. Well, most bacteria reproduce by fission. One bacterial cell divides itself in half, you get a full bacteria. Again, millions of different types of bacteria, and they all reproduce like that. That's not going to do much for them. So we have to get to some other criteria. There's our one cell undergoing fission, two cells. Mostly they do that. Another way to categorize, and this becomes more applicable when we start talking about prokaryotic organisms, bacteria and archaea, we can look at categorizing them based on their nutrition and their metabolism, assuming we know something about their nutrition and their metabolism. Now, we've talked about nutrition in the terms of the media that are used to support the growth of bacteria. We said you've got to have a carbon source, a nitrogen source, you have to have water, you have to have certain minerals, you have to have maybe coenzymes, et cetera, et cetera. when you're dealing with nutritional categories for the taxonomic scheme of things, we divide these based on what the organisms use for carbon and what they use for energy. We know they have to have nitrogen, but that doesn't usually get incorporated into the naming system. I'm not really sure why. So we divide them, or we set up these criteria. The criteria again have to do with where they get their energy and where they get their carbon. They can get energy from light or from chemicals. So again, think small. Think about bacteria. Bacteria as a huge group can get their energy either from light or from chemicals. Where do we get our energy? Chemicals. We have to take in organic compounds and break them apart. So we get our energy from chemicals. You can lay in the sun and get warm, but you can't do photophosphorylation. You can't make ATP using light. There's quite a bit of evidence actually to show that microorganisms grow in association with thermal vents on the seafloor, but I've never seen anything that says they're actually using the heat energy to make ATP. So as far as I know, light or chemicals. Carbon comes from either an inorganic source, and our inorganic carbon compounds, remember, were carbon monoxide, carbon dioxide, and bicarbonate. Or carbon can come from some kind of organic compound. Well, okay, so we've got energy, we've got carbon, and of course we put names on things because it's biology. So there's specific terms that apply. We put terms on everything. The end part of the word, phototroph, troph can mean feeding or activity, activating. It has to do with what organisms use for nutrition or activation. If organisms are phototrophs, photo means light. This means they get their energy from light. Certain organisms are phototrophs. We are not. If organisms are chemotrophs, it means they get their energy from chemicals. Then we consider carbon source. If they can use an inorganic carbon source, if they can pull carbon dioxide from the atmosphere, or if they could use bicarbonate from water, we say they are autotrophs. Auto means self. It means they can pull inorganic compounds, fix the carbon, and make their own organic compounds. And if they are reliant upon preformed organic compounds, we call them heterotrophs. We've got these four categories based on the first two are energy source, the second two are carbon source. Living organisms have to have both. They have to have an energy source and a carbon source. So we reorganize these, put them together to come up with four nutritional categories. The first category is photoautotroph. Photoautotrophs get their energy from light, and they can use inorganic carbon as their carbon source. What kind of organisms are you familiar with that are photoautotrophs? Plants. Look outside, you see green things out there. Trees, grass, etc. They're green, they use the light-trapping green pigments, the chlorophylls. They use light to make ATP, and then they can pull CO2 from the atmosphere and use it to make sugar. So photoautotrophs, plants. Chiefly plants, but it turns out there's quite a lot of bacteria that are also photoautotrophs. Cyanobacteria, for example. Algae are photoautotrophs, and technically they're protista now, not plants. If we have organisms that are using light energy, but they use organic compounds for carbon, we call them photoheterotrophs. As far as I know, the only organisms that fall into this nutritional category are prokaryotic. So there are some prokaryotes. They can use light energy, but they have to have organic carbon. One of our morphological unknowns, in fact, it's the only one we have that is a spirillum. They are photoheterotrophs. They can also function as chemoheterotrophs. Then we have chemoautotrophs. Chemoautotrophs use chemicals as their energy source, but they can use strictly inorganic chemicals, and they use inorganic carbon. A lot of these organisms live in some very unusual places. This is something that people are just kind of finding out about. Bacteria can live inside rock, inside layers of rock, really deep in the mud. What are they doing in there? Well, they're functioning using inorganic compounds, strictly inorganic compounds for both energy and carbon. Again, these are all prokaryotic as far as I know. So the prokaryotic organisms are a little strange. And then we have organisms that are chemoheterotrophs. Chemoheterotrophs are using chemicals for energy, and they're using preformed organic compounds as their carbon source. Turns out they're actually using preformed organic compounds for both carbon and energy. And who fits into this category? We do. All animals do. Fungi do, and protozoa do. And some bacteria. So this is a really big category. All types of animals, fungi, protozoa, multicellular parasites, animals, and I think I said that. A lot of bacteria and archaea are also chemoheterotrophs. Because this is such a big category, but probably more importantly because we are in it, biologists have subcategorized chemoheterotrophs. So we have some subcategories. If chemoheterotrophs are saprotrophs, these used to be called saprophytes. Now they're called saprotrophs. Sapros means rotten. So these are organisms that use dead and decaying organic material. We don't like to think of ourselves as saprotrophs, but how many of you eat yogurt?
Yes.
Yeah. Well, don't look now, but it is partially decayed because some bacteria were eating it first. If you eat cheese like brie that has that nice fungus mycelium on the surface, they were eating it first. So we do tend to eat some partially decayed food material. Anyway, saprotrophs are using decaying organic material. We often also, you'll hear these guys referred to as decomposers. They tend to decompose organic material. We do that too. Anyway, fungi, a lot of bacteria, a lot of animals too. Then there's another category, parasites. Parasites are organisms that use some other organism as their source of nutrition. So they get their carbon and their energy from somebody else. Sometimes they live inside the host, sometimes they live outside the host. So here we see some tapeworms. Tapeworms are parasites that live inside the host. You've all encountered mosquitoes, surely. Mosquitoes, lady mosquitoes, are ectoparasites. They live outside their host and they have to attach to the host and take a blood meal. Apparently male mosquitoes suck plant juice. Another term that's a subcategory of parasite is hypotroph. A hypotroph is an obligate intracellular parasite. That means something that can only grow and reproduce inside a host cell. A tapeworm is not a hypotroph, they're too big. So hypotrophs typically are single-celled organisms. As soon as I say that, I think, yeah, but if we wanted to categorize viruses, that's where they would go. Viruses are hypotrophs. They can only reproduce inside of a cell. So all hypotrophs are parasites, but not all parasites are hypotrophs. You're going to see this kind of logic several times. Okay, and then in ecology textbooks, you'll hear these kinds of terms: carnivore, herbivore, omnivore. This has to do with what kinds of food material, what kind of organic material the organism is breaking down. If an organism is a carnivore, it eats meat. If an organism is an herbivore, it eats vegetation, plant material. If an organism eats anything it can get its hands on, it's called an omnivore. We're omnivores, which is one of the reasons we're so successful as organisms. We'll eat just about anything. There are lots of organisms that have a very restricted diet, so if you take away their food source, they're going to die. You take away our food source, we'll go find a different one. It's like, okay, you won't let me eat that, I'll eat something else. We're very versatile. Okay, so that's just nutrition. Nutritional categories. Again, 4 basic nutritional categories based on both carbon source and energy source. Now we can look at metabolism. We're going to go into metabolism at length when we get to that section during the semester, so this is a real quick and dirty coverage of metabolism. We can divide metabolism into 2 categories. We say organisms are either respiratory or they are fermentative. Which are we? Respiratory. We are, but what happens when you go out and you exercise strenuously and the next day your muscles hurt? What were your muscle cells doing? They were fermenting. So we call ourselves respiratory, but our cells can run a fermentative metabolism. When they do, they produce lactic acid, which tends to be toxic and causes our muscles to hurt. This unfortunately tends to happen quite a bit when you start looking at living organisms. We try to put them in discrete categories and they sneak out and kind of slide into another category too. Nevertheless, we're going to say, okay, we've got respiratory or fermentative and say that things fall into either or. So what makes an organism respiratory? A respiratory organism is one that's capable of using an inorganic compound such as molecular oxygen, O2, as a final electron acceptor. It just so happens we do this. If you don't believe me, try putting a plastic bag over your head. We use molecular oxygen. What do we use it for? It's a final electron acceptor. How many of you know what I'm talking about? A few. Okay, so you're gonna have to get a handle on this final electron acceptor business. Basically, molecular oxygen picks up electrons and within our cells becomes water. That's what we do with it. And the picking up of the electrons is very, very important because it allows coenzymes to function, which allows us to make ATP, etc., etc. Anyway, just remember respiratory organisms use an inorganic compound as a final electron acceptor. And even though that doesn't make sense at the electron acceptor level, hopefully it will later. If organisms are fermentative, they use organic compounds as their final electron acceptor. The other thing is, respiratory organisms, their electron acceptor typically comes in from outside, so oxygen diffuses into cells, whereas the fermentative organisms very often use pyruvic acid as their final electron acceptor, and they make it. So it's made inside. It's not something they have to import. So it's all about electron acceptors. And as I said, if you're not familiar with this electron acceptor concept, we'll get there. Don't worry about it too much right now. Oh, and this is talking about reduction. Oxidation reduction. How many of you like oxidation reduction? Well, you need to know, when molecules pick up electrons, they become reduced. The easiest way to think about it is think of those electrons as a minus sign, and negative numbers are smaller. Otherwise, it doesn't make sense because the molecules, when they pick up electrons, they also pick up hydrogen, and so they actually get bigger, and then it's like, wait, that's reduced. It doesn't have to do with size. Okay, so we've got nutrition and metabolism. The next thing we can consider is, can we categorize things based on their gas requirements? So again, a whole bunch of terms. If organisms require oxygen, and they require it for their metabolic processes, we call them obligately aerobic. We are obligately aerobic. And the reason we are is because we have a respiratory metabolism. We use molecular oxygen as a final electron acceptor. So obligate aerobic organisms require oxygen. There are some bacteria that are obligately anaerobic, which means oxygen is actually toxic to them. If you expose them to oxygen, they die. So they don't want to have anything to do with oxygen. They're obligately anaerobic. And then there are many of them that fall into this category in between. They used to be called facultatively anaerobic. Now you'll see textbooks where it's either facultatively anaerobic or aerobic. If they're facultative, it means they can go either way. They can live with or without oxygen. If it's available, they can run a respiratory metabolism. If it isn't, they ferment. Well, some of our cells are like that too. Some of our cells are facultative. Mostly they're respiratory, but under certain circumstances they ferment. So it's not too unusual. And then there are some bacteria that are called microaerophils. These organisms live in environments with low levels of oxygen. And often, when they are in those environments with low levels of oxygen, they're using oxygen as an electron acceptor. Consequently, they take it away from the environment, and they make the environment more anaerobic, and that allows the anaerobes to survive, the obligate anaerobes. So, this is talking about looking at broth media. Sometimes you can kind of get a hint what kind of gas requirements organisms have by looking at how they grow in a broth medium. If they are obligate aerobes, oftentimes they'll form a structure called a pellicle. So they'll grow in a layer right on the top. Although if you look at this, you can see there's growth down underneath too, because there's a lot of turbidity in that medium. But obligate aerobes will often form a pellicle. They'll sit right on top. If they were obligately anaerobic, they might sit on the bottom. They're facultative, they can grow all over the place. Here we see our anaerobes. As soon as I show you that, I think, well, that might just mean they're not modal. And any of you looked at our morphological unknown tubes, almost all of those cultures are sitting on the bottom. It's not because they're anaerobes, it's because they can't swim. And gravity causes them to sink. We sit on the bottom. Another thing, this is mentioning there are some bacteria that textbooks will tell you are obligate aerobes, when in fact they are not. One that comes to mind is Pseudomonas aeruginosa. Pseudomonas aeruginosa, you'll often hear it described as being obligately aerobic. They are respiratory, but they are not obligately aerobic because they don't have to use oxygen. Remember, a respiratory organism uses something inorganic as a final electron acceptor. Pseudomonas can use nitrate. So if there's no oxygen available, they can still grow if there's nitrate. And so sometimes Pseudomonas, which is a significant human pathogen, will show up growing in an anaerobic environment with Clostridium. It causes people go into confusion. It's like, how can they do that? Well, they aren't really obligate aerobes. They're just obligately respiratory. So there we see Pseudomonas aeruginosa. These guys produce a green pigment. It's water soluble and it's diffusing into the medium. That's a hint to somebody with their morphological unknown. Okay, next set of criteria, temperature requirements. So again, we have these categories. If organisms like very cold temperatures, we call them psychrophiles, not psycho, psychro. Psychro means cold. So really cold, -5 to 20 degrees centigrade. -5 is really cold. In this image, what you're seeing is a snowfield. You'll notice the blue color from water, but you see the pink coloration? That's called watermelon snow. How many of you have ever seen watermelon snow? If you hike in the Sierra Nevada in the summertime, a lot of times you'll see watermelon snow. Don't eat it because it has some type of psychrophilic algae in it and it can make you sick. Don't eat yellow snow. That's different. If organisms like a warm environment, we call them thermophiles. Warm as in 45 to 60 degrees centigrade. 45 degrees centigrade is going to feel hot. So when we say warm, really warm, and 60 degrees, you can't put your hand in. So these would like, what you see here is a hot spring. So there's got to be something in between, which there is. Mesophiles. Mesophiles are organisms that like moderate temperatures. By the way, remember, phil, P-H-I-L, means loving. So these organisms actually grow best at these specific temperatures. Mesophiles grow best between 20 and 45 degrees. So where might you find them? Hint, look at the picture. In your sinuses, inside my stomach. Inside and in association with you. Our normal body temperature is 37 degrees centigrade. And consequently, the organisms that live with us are mesophiles. Most of the organisms that would be pathogens are mesophiles. And then there are some organisms that are called hyperthermophiles. There's a couple of other terms you might see applied to this. Hyperthermophiles really like it hot. So they're going to grow in temperatures that are above 60 degrees, sometimes right up almost 100 degrees. 100 is boiling. They'll be just a little bit less than that. So these guys would live in hot springs where the water might be boiling in one region and they're living very close to that. They also show up on the seafloor where the hot water vents exist. They like it hot. Then we have the term psychroduric. Psychro again refers to cold, but duric means to endure. If organisms are psychroduric, it means they're not growing best in cold temperatures, but they can endure exposure to cold. And when you're dealing with prokaryotic organisms, they are almost all psychroduric to the extreme. Bacteria are maintained in freezers. A lot of living organisms, a lot of people have collections of living organisms. And plants, they press them and they're dried up. Animals, they stuff them and they hang them on walls. Bacteria, they freeze them. and they put them into these freezers. The one that we have in the back is a -70. Actually, it's supposed to range, it's supposed to between 68 and 72. Right now, it's been running at 68 because it's right on its last legs. But some of those freezers are -90. That's the temperature of liquid nitrogen. It's cold. And the bacteria just hang out there at that temperature. They don't die. They are psychroduric. This is important to know so that if you think freezing will kill bacteria, think again, because it absolutely does not. They're psychroduric. Some of them are thermoduric. Thermoduric means they can tolerate or endure exposure to heat. What did we just learn about? Actually, last time you got this as the last, or the category of specialized cell types that can tolerate exposure to heat. And we did the pasteurized soil things. Endospores, and exospores are thermoduric. So there's some endospores. Tonight we're going to be doing endospore stings, so you get to see. Hope you get to see them. Okay, another way to categorize acidity versus alkalinity, or pH requirement. The good news in here is you guys don't have to calculate. You're not going to do anything numerical with pH, but we do use a lot of pH indicators. And I talked about this relative to media. pH can be maintained in a medium using buffers. Buffers are substances that resist pH change. This is because a lot of times bacteria like to have a pH that's pretty neutral. They want to be right around 7, which is neutral pH. So maybe between 6.8 and 7.2. If you know they can't tolerate exposure to acid or alkaline environments, then you put buffers with them to prevent them from changing their pH. If you want them to change the pH of the environment and you want to see that pH change happen, you put pH indicators in there. So these are substances that change color in response to changes in pH. There are some bacteria that, and some archaea also, that are acidophiles. Acidophiles are organisms that really like acid environments. And they can live in some very strange environments, places where you wouldn't think it would be possible for anything to live, exceedingly acid environments. There are also some that live in alkaline environments, and they are called basophiles, but I'm not going to use that term because you know what a basophil is?
White blood cell.
It's a white blood cell. It's a category of white blood cells. So I'm just going to eliminate confusion. Never mind, we're not going to basophile. This is showing some organisms that are acidophiles, and they also like hot environments. So they're thermoacidophiles. They like it hot, and they like it acidic. Yeah, never mind the basophiles. We're not going there. Okay, another way to categorize osmotic pressure requirements. You need to know about those, the different forms of environments, whether they're isotonic, hypertonic, hypotonic, and we went through that already. That has to do with effective osmotic pressure or tonicity. If they're isotonic, then the solute concentration is the same or similar to protoplasm. If it's a hypotonic environment, hypo means less than or lower, and so the solute content is what's low. So you have more water. Fresh water is very hypotonic. And if the environment is hypertonic, then you have high solute. So salt or sugar. Hypertonic environments I mentioned earlier are used for preservation because they tend to cause cells to lose water. Water goes out and the cells can't carry out their metabolic processes. Oh, I just told you that. Hypertonic environments often inhibit microbial growth. Why? Because they cause water to exit. Without water, the cells can't carry out the metabolism. They have to have water for their hydrolysis and reactions. There are some organisms that grow very well in high salt. Some bacteria, these are called halophiles. There are regions in San Francisco Bay. I don't know how familiar you are with San Francisco Bay, but that's an aerial photograph looking down on the salt production plants, where they basically evaporate seawater, and the organisms that are living in there are called Halobacterium holobium, and they are halophiles. They like exceedingly hot or exceedingly salty water, and they are pigmented pink. And then another way to categorize organisms is based on their environmental relationships. This is an area where new investigations are just getting going. It's been really hard to study environmental relationships with bacteria when most of what we know about bacteria we learned by taking one type, putting it on media all by itself, growing it as a pure culture and saying, here, you have to function this way. That's not normal for them. It's not normal at all. And we now know that most bacteria will not grow that way. This is why what we know about them is rather limited. So environmental relationships have become very important. Symbiosis, I mentioned this term earlier. Symbiosis is the condition or situation where you have two or more different types of organisms living together in a close association. In biology, it used to be thought of as a rarity. It's like there'd be one or two examples that would be pointed out. It's like, look, this is symbiosis. We now know every eukaryotic organism is a symbiote. Every single one. Eukaryotic organisms don't exist without prokaryotes. Nevertheless, we can divide symbiotic relationships into categories as well. Some organisms are pathogens. A pathogen is something that if it gets into a host organism is likely to cause disease. For a long time, that was the only interest people had in bacteria. Once Robert Koch established, hey, some of these can cause disease, it was like, whoa, they all cause disease, we've got to kill them. We now know that's not true. Some organisms are parasites. We will talk about some specific parasites. That's a symbiotic relationship where one benefits the parasite and the host is damaged. Typically, parasites don't damage their host too much because if they do, the host dies and then the parasite dies too. And then there's a mutualistic relationship, mutualism, where everybody benefits. Apparently, this is more the norm. We have different organisms living symbiotically and they're actually helping each other out. We now know that our normal microbiota is helping us. And we're just beginning to find out what else are they doing, because apparently the organisms living in our gut influence our immune system, they influence our behavior, they may influence disease processes like diabetes, maybe autism. We don't know. We don't know what those organisms are doing. We're trying to find out. So this is an area of intensive research. Not just, again, pathogens infect something, parasites use a host as food. Mutual, beneficial for everybody involved. One common symbiotic relationship that's often talked about in biology textbooks is the lichen. Because we see these little things. How many of you know what a lichen is? Okay, so well anyway, there used to be lichens growing on the rocks, on the wall inside the campus center. It was because they were growing on the rocks before the rocks were put in the wall, and they just stayed there. You find lichens on rocks, on soil, on tombstones a lot. They're these little funny looking patches. Well, it turns out a lichen is actually a symbiote. It's made up of fungi living symbiotically either with algae or cyanobacteria. There are bacteria that live in association with plant roots. They form these nodules. A lot of times people think that there are certain plants that can fix nitrogen. As far as I know, no plant can fix nitrogen. Farmers grow nitrogen-fixing plants like peanuts and clover and vetch, etc. Those plants can't fix nitrogen, but there are bacteria living with their root nodules, and the bacteria can. The bacteria can fix nitrogen. And they don't have to have heterocysts to do it. So again, mutualism, beneficial for everybody. This is just showing the, if you go to the ASM website, you can find their little fact sheet on the human microbiome. And again, this is something we're interested in. Our bacteria benefit us, we just don't know how. Be nice to your microbiome. Okay, then another way to categorize, especially bacteria, is biochemical analysis. Not surprisingly, the more we've learned about chemistry and microbial metabolism, the more we can apply this biochemical analysis. There's different categories. One, enzymatic testing. In this laboratory, you're going to do an assignment called Physiological Unknown 1. And Physiological Unknown 1 is based entirely on identifying organisms using enzymatic testing. So you're going to learn about this. You put them on different media to see what kind of enzymes they can make. Can they make citrate permease? Can they make urease? Can they make tryptophanase? Those kind of things. So enzymatic testing, you'll get to learn more about that. Chromatography is used extensively for categorizing things, but not in this lab because we don't have a chromatographer. So, yeah, it exists, we don't do it. Serological testing, or serological typing. This is a type of biochemical test that involves using antibodies to either identify specific types of microorganisms, mostly, or to detect them, to determine if they're there. So what are antibodies? They're proteins. We produce antibodies. They're part of our immune system. Our antibodies are very specific in terms of what kinds of antigens they will bind with. When you're talking about serological testing, the antigen is some kind of microorganism. It can be bacteria, it could be fungi, it could be protozoa. But if you have an antibody, it's very specific. If it binds with an antigen, then you know what the antigen is. So serological typing, you use known types of antibodies to identify some unknown antigen or to detect it. You know how animals are tested to see if they have rabies? Well, they have to take their brains out, and they take some of that brain tissue, and they use fluorescent antibodies that will bind with the rabies virus if it's there. So you have to sacrifice the animal, it's not nice. You have a pet, immunize it. Get those rabies vaccines. Okay. Another biochemical analysis that is only useful for the identification of bacteria is called phage typing. A phage, the term phage is short for bacteriophage, and bacteriophage is a type of virus that can only infect bacteria. It's kind of like serological typing in that you use known types of phage. They will only infect specific bacteria. They're very host specific. So if you know what kind of phage you've got and it infects something, then you know what that something is. This is something we'll be doing in the laboratory. You'll get to see phage typing firsthand. Kind of simplistic, but anyway. So then you get to nucleic acid analysis. And this is really the nitty-gritty because nucleotide sequence is going to tell you everything the cell can do. Kind of in a roundabout way. But anyway, nucleic acid analysis. Nucleic acids are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. There are different ways of looking at these. For a long time, people were looking at percent base composition. Remember the bases, adenine, thymine, cytosine, guanine. They looked at them in terms of the pairs, so how much adenine, thymine was there, or how much cytosine, guanine was there. It turned out this was useful for telling you if organisms were not related, but not terribly useful for telling you if they were. They can have exactly the same percent base composition and not be related. Hybridization involves putting two nucleotide strands together with hydrogen bonding. DNA, two strands come in the hydrogen bond. They can only do that if the nucleotide sequences are complementary. So you can, if you have one piece of DNA that you know the nucleotide sequence for, you can determine how related it is to another piece by how much of the hydrogen bar. We use hybridization when we do the PCR because we use primers that have to hybridize with DNA to get the PCR to work. By the way, how many of you know what PCR is? So PCR stands for polymerase chain reaction, and it's a method used to amplify DNA. We're going to spend a lot of time talking about PCR because we use the PCR for physiological unknown too. We have to amplify the 16S ribosomal RNA so that we can nucleotide sequence that and find out how our bacteria relate to other bacteria. Gel electrophoresis, this is another technique that we use. DNA has a charge, so if you put it into an agarose gel and you establish an electric gradient across that gel, the DNA will move and it'll separate on the basis of size. DNA fingerprinting involves electrophoresis, at least sometimes. We're going to do something like DNA fingerprinting. In this lab, we're going to call it restriction fragment length polymorphism, or RFLP. And then the last one, nucleotide sequencing. As I said, knowing the sequence of bases, the sequence of nucleotides, that really tells you what an organism is in terms of its relatedness. Our physiological unknown 2, the identification is based almost entirely on nucleotide sequence. This is significant because it's changing how people function in a clinical setting. So all this business we're learning about gram stain, and then tonight we're doing acid fast stain, and then we'll do capsule stain and so forth. All that morphology business and even enzymatic testing is eventually going to be replaced by nucleotide sequencing. Because with nucleotide sequencing, you can get an absolute identification very quickly. It involves computer technology. But there are some rapid nucleotide sequencing systems that are set up where you can get answers for identification within a couple of hours, pretty quick. Next generation sequencing, this is something that is being developed, and this is looking at sequences not of individual cultures, but sequences of communities. So they're looking at studying organisms in place in natural communities, and it has gotten really crazy. If you're not familiar with the term genome, the genome is the total DNA content of an organism. Chromosomes of an organism, it doesn't have plasmids. And so genomics is the study of all the genomes of all the organisms that have been identified. And it all, again, it involves computer technology, comparing genomes. The, what's called metagenomics, they're only looking at parts of genomes. They're looking at specific little parts. And again, analyzing communities in place. So they're looking at a variety of different environments and figuring out who's related to who. This other thing that's talked about, ribosomal RNA genes, ribosomal multilocus sequence typing, that has to do with identification based not only on ribosomal RNA nucleotide sequences, but looking at all the proteins associated with ribosomes. Ribosomes, again, are really important because cells can't function without them. Well, there's also analysis of proteins, which we don't do at all. There's analysis of fatty acids, et cetera, et cetera. We're running out of time and we don't do that anymore. So what do you do with these criteria? Well, you can use dichotomous keys where you ask one question, you have two separate answers, you either get A or B, and then depending on which answer you get more questions and more answers. There's a kind of a dichotomous key in the lab manual. Or you can do cladograms, where you take one feature and you kind of compare it across the board. So again, a dichotomous key, you're basing your identification on a series of choices between alternate characters, A or B, or 1 or 2. And you have to ask a whole series of questions. Is it gram-positive versus gram-negative? Is it cocci versus bacilli? Is it single cells or clusters, et cetera, et cetera. Cladograms are these branching tree-like patterns that show relationships between organisms. We won't necessarily be looking at a lot of those, but that's an example. That's a really simple cladogram. And if you have a very complex one, it's called a phylogenetic tree. And here's one. This is a phylogenetic tree based on ribosomal RNA nucleotide sequence. But it says you've got bacteria, you've got archaea, and you've got eukarya. There's a newer version of this that shows the bacteria category huge, just huge. This one would suggest that the bacteria and archaea and eukarya are all equally represented on the planet. It's like, no, they're not. Bacteria, that category is immense. We just don't know anything about it. It's kind of humbling. Anyway, so taxonomy is changing, new stuff being found all the time. And yeah, mostly you're just looking at some terms. And again, phylogeny is evolutionary history. This also shows there was a lot of sharing. All eukaryotic organisms came from sharing between the two prokaryotic groups, the bacteria and the archaea. There's another look at the tree of life. There's a lot of different versions of this. Notice this is an egocentric version because it looks like it starts out and culminates with humans at one end. It's like we are not the most important entities. Oh, so that's nice. this is also significant. Apparently, there's more things that have gone extinct, disappeared, than there are the things that exist. We tend to get all excited about things going extinct, but it happens. Lots of things have gone extinct. And we are not immune to that process. Never mind. Okay. Yeah, so there's a lot of differences, a lot of changes. Probably the most significant feature of that is if you go to a different class, you will see something else. You'll see a different set of criteria. You'll see different categories. A lot of biology classes don't do the domain thing at all. I think that's okay. So when you come back, your lab quiz covers pure culture technique, which I kind of forgot about, but the night that we did direct and indirect staining, we also did pure culture technique. We're still working on pure culture technique, because only a few people have turned in their properly streaked pure culture plates, but that's part of the quiz. So pure culture technique, indirect stain, direct stain, ram stain, and KOH.
Yeah, that was it. Yeah. When do we have to do that?
Let's start at 5 after, because I kind of went over. I kept wanting this thing to stop. Let me wash this off, okay? Because I don't know how to do it. Too much. So when you pick it up initially, make sure you just touch it. Don't pick up a big boom of glass. Touch. Because remember when we first streaked, we were using raw and you couldn't see anything. So you want a very small amount. Your pattern is good. You're seeing too much solid material. Oh yeah, yeah. There's no hurry on it. I want you to feel comfortable with doing it, so you can feel when you're doing it right. There. Well, and we've also got an air contaminant in there. Although, I said air contaminant, that might be the bacillus. So I'd do it again because it's not pure. Whichever. Okay. There you go. So, one of the things that we're supposed to do for the morphological unknown is colony size. So you want to measure that. That is your colony. Yeah, so you would say that's how big the colony gets in 40 hours. Because the next time you see that plate, they're going to be right out to the edge. They would keep going except they run out of food. Yeah, they have a very distinctive looking colony form too.